hello there. We will be talking about my short movie that I made, or thing, The Journey of Protein Synthesis. If you haven't watched that, you probably shouldn't. It's absolutely chaotic. Anyways, I am here to explain it. So, to start from the beginning, DNA strand. Node. Thank you. So, to start off, here are some things that you should probably know. We have DNA. We have RNA. DNA, you should probably know if you've ever taken any biology class. 2 strands, nitrogen bases. RNA, 1 strand, many nitrogen bases. Anyways, so the nitrogen bases are thymine, adenine, guanine, and cytosine. For RNA, thymine is actually replaced with uracil. We'll get to that later. For RNA, there are 3 types. We have tRNA, mRNA, and rRNA. Okay? So for tRNA, transfer RNA, they will bring amino acids to a ribosome. We'll get to that. Messenger RNA. That is that. This guy in this movie is an mRNA. Oh yeah, also, you will see on this guy, there are blue, this is supposed to be yellow, but it looks green. Blue, yellow, red. So, notice this, if you were to count out this specific sequence. It is the exact same as this, except the blue, or thymine, is replaced with purple, uracil. I'll get to how that happens a little bit. Oh gosh, I know. Oh gosh, no. So flippin'. Anyways, so let me repeat myself. Anyways, blue, thymine, yellow, adenine. They pair up together. Pair. Like I said, you get this idea. Count it out. It is the exact same as the one on this guy that is played by yours truly, except that the blue is replaced with purple, uracil. Blue is replaced with purple. Yeah, you get it? I think you get it. So, what is the purpose of an mRNA? An mRNA is made, which is coated with the nitrogen bases that I mentioned earlier, which will be translated into amino acids, which then will form polypeptide chains that will fold together to make proteins. Okay, follow me. Anyways. Alright, so for protein synthesis, there are 2 processes. We have transcription and translation, okay? Transcription makes RNA, which, translation makes the proteins from the RNA, from the messenger RNA. So how transcription works is that an RNA polymerase attaches onto an RNA, moves up, finds nitrogen bases that pair up with these, and then make an RNA strand that comes off of it. which is shown through this. This machine right here is actually called RNA polymerase. This is just the best way I could represent it. Anyways. So, yeah, that's essentially how it's made. So, if we go, sorry, back to this, it might be a little disorienting. In the story, he meets a fellow tRNA. I will get to him later. And then, meets a security guy. This guy obviously doesn't exist within cells. I do not need to explain that. And he talks about how there are introns and extrons that need to, but introns that need to be cut out. They are essentially nitrogen bases that are essentially useless for making a very certain protein. So, what happens is that they are cut out, and you can see that he is significantly shorter than he was. He's also given a cap and tail. I'll also explain that. So what happens in this is that there are certain sections that are just useless, get erased, and then the extrons that are then spliced back together to make it, which that is why it's shorter. And then for an RNA to be able to signal which end is the front, which end is the back, it is given a cap and a tail. So now, they leave the nucleus to they leave the nucleus, enter the cytoplasm, which then there's this Godzilla mutated protein. We'll get to him. One epic fight later, he dies. Anyways, so now we come to the ribosome. The ribosome is very important. It is pretty much the main component of translation. Alright, so I'll get to this better view. Anyways, so to understand translation, we need to understand what a codon is. If I can, this is a codon. Codon is a set of 3 nitrogen bases that code for a certain amino acid. Amino acids are stringed together, form a polypeptide chain, which then is folded to make a protein. So, this is where our fellow tRNA comes in is a very important part. They have the opposite of this. This is not the opposite. I just put a random thing. This is the opposite of a codon, which is called an anticodon. So, an anticodon would be able to connect to a certain part of an RNA strand that is the codon. So this sequence would match with this sequence. This sequence is A. Hold up, wait a minute, I got this wrong. A G A would be the same as U C U. Okay. Anyways, so in the ribosome, it is mainly consisted of 2 parts. We have the the larger subunit and the smaller subunit. They just kind of sandwich the RNA like this, which then kind of runs it like a feed tape, sort of something like that. Anyways, so tRNAs come in, link up to connect with their codon parts. Oh, okay. Let me explain. There are 20 amino acids. Okay, there are 4 nitrogen bases for 3. So that would make 64 possible combinations. So a lot of it is actually redundant for a few amino acids. Like there's probably like 4 sequences for like one amino acid. So, hold up. Anyways, yes, matches with their... Hold up, where was I? Yeah, matches up here. And the the tRNA that's already in here will put its amino acid chain, connect it to the one over here, and then this one will get ejected out, okay? And then the entire RNA system will move forward. This is represented by just a general belt, explained by Mr. Meiosis himself. Oh yeah, let me also explain something. Hold up. Hold up. This is really... I can make this bigger. Still make this bigger. Hold up. There we go. Anyway, so you see this protein folder. That's not actually in the ribosome. I just made that for plot reasons. So, when these fellow tRNAs walk into the thing, they have a certain code which you can actually see on their Minecraft skins. So, for with the certain colors that are represented, G is represented with orange. Her name is CCC, which the anticodon sequence is GGG. Guck is CAG. So that's a very interesting way for me to be able to do it. So now time for our secondary main character that we meet all the way back at here. AUG, AUG, is actually the most common RNA starting sequence, which the anticodon is UAC. Yeah, UAC. So, this guy, him dying is, makes it so that he can't do it, so he can't make his thing, so, all chaos. He comes back. Day is saved. They make their protein.
The sequence for
So, hold up, let me go back. So yes, should understand this, right? So, once the RNA is ended, the polypeptide chain will release, and then another part of a cell will fold it to make a protein, which will call a protein enzyme, something like that. So then it'll actually do its actual job. So this is hold up. So now it's time to talk about mutations. So our fellow AUG fights Godzilla, or just a mutant protein. So how mutations happen? And so if, let's say, one of our fellow mRNA has you flipped out a singular, let's say, cytosine with a uracil, that will cause an amino acid to be different, which then would cause the protein to work differently in certain cases. But that is only what happens if you replace one. Let me go into a great example if you add or remove one. So, let's write 3 letters. We have we have U, you saw the dog. Let's say that. You saw the dog. Okay, what I mean by saw is that you see it. Now, let's add a singular letter. So, let's go add a B. And remember, everything is read in threes, okay? So, that turns from you saw the dog to this. BYO, USA, what the hell, Edo, and G. See where this can become a problem. So, let's say if you want to make an amino acid that, let's say, makes a certain process go faster. And let's say that there's a mutation in your DNA which causes a singular thing to be added. Although this is a very, very, very dramatic case. No, stop it. Although this is a very dramatic case, and just for plot, we have Godzilla, who is mutated. This is also what is the main... Yeah, see, cool mutant, see? Mainly for the funny, but this won't happen. Don't worry, you shouldn't need to worry about proteins shooting radioactive beams across everything. That's not how it happens. So, that should conclude all what you need to know about protein synthesis, transcription, to translation. DNA, RNA, here's very, very important, okay? So, I'm tired. So, we're now done, okay? You can leave. I'll be waiting. You leave. I'll end the video after you leave, because, yes. Have you left it? Have you left the video yet? No? I'll be waiting. You still haven't ended the video? Yeah, fine. I'm fine. I'll just nuke you. Fuck.